Morning and very welcome to my lecture. Today we are going to do biology chapter 1 of class 12. We will learn about sexual reproduction in flowering plants. We will go through the chapter main points and see what are there. So in this chapter you will learn about male reproductive structure in flower or stamen, that is about the flower. You will learn about pollen grain, which is also known as microsporangia or microspore. We will learn about that, and that is called male gamete. We will learn about female reproductive structure in flower. We will learn about ovary, ovule, or megaspore or mega female gamete structure present in the flower. We will learn about pollination. We will see what outbreeding is. We will see what artificial hybridization is. We will discuss about fertilization and post fertilization events which leads to formation of fruits from the flower. Apomixis and polyembryony will also be discussed in this chapter. So let's begin. In class 11, you have read about kingdom Plantae, which have 5 subclasses: algae, bryophytes, pteridophytes, gymnosperm, and angiosperm. In these categories, the angiosperm is known as flowering plants, that is, those plants which have flowers in them. You have seen flowering plants, you have seen special characters of flowering plants, special structures of flowering plants which are different from non-flowering plants. Okay, so in a plant there is flower, and in flower sorry okay, in flower there are typically parts of a typical flower. The parts of a typical flower consist of stamen, petal. Okay, the essential parts of a flower are androecium and gynoecium. That is, the androecium is the male part and the gynoecium is the female part. The non-essential part, which are not essential, are petals and sepal. The group of petals is known as corolla, and group of sepals supporting the flower is known as calyx. Stamen or male reproductive structure. Stamen, see, there are number of sticks present in flowers, so you can differentiate between those sticks, and there are two types of stick. One stick is thicker than other, and the other sticks are present uh, around that central stick. As you can see in gray color, there is the stick is thicker than other sticks present in green color, green and orange color. The orange colors stick are comparatively thinner than the gray color stick, isn't it? So say for example, this is a flower of hibiscus. So in hibiscus plant you will see small and thin green colored sticks, and above the stick the orange color uh, oval like structure is present, which is known as stamen. And above stamen, inside the stamen, there is present anthers. You must have seen that yellow color powdery dusty pollen grains present. So these green stick and the powdery structures present on the tip of that is the male reproductive part, isn't it? The stamen or male reproductive part. So the tip is known as anther and the stick is known as filament in biological terms, and these both two forms the male reproductive part, or also known as stamen. And inside stamen is present pollen grain. We will also see that. So when we cut the anther in between from the between we make a transverse section, we cut a transverse section of the anther. We say this four lobed structure, okay, two lobed structure and four circles uh, on each corner of the anther, isn't it? Once again, let's see, when we again the anther is cut transversely, transverse section of the anther, we see four corners and it is bilobed, two lobed is present, and the four centers, the circles present in the four corners is known as microsporangia. You say it microsporangia or you say it sporogenous tissue, both are similar terms applicable in different situations. So this anther is bilobed and diathecous. Each lobe has two theca. Each lobe, the you can see it is clearly distinguished in two lobes, and each lobe has two microsporangium. Okay, and the that is, you say it microsporangia or two theca. That is why it is known as diathecous. Again, we see anther in its thecus there is number of tissues present, isn't it? So these will be asked in exam and you must know about this. This will be asked in structure of anther. The outer layer is known as epidermis. Everywhere in plant the epidermis is always the outer layer. Inside the epidermis is endothecium. Inside endothecium there is middle layer, and just below middle layer there is tapetum. You must remember what tapetum is. Tapetum is nothing but the food for the pollen grain, future pollen grain, isn't it? And the sporogenous tissues are tissue from which the pollen grain will be made. So we have just seen the structure of microsporangia. You say it microsporangia, you say it anther, you say it diathecous, you say it pollen grain. We will see about structure. They are all name of the same thing, isn't it? Name of the same thing present in different times. At different times of the development, the name changes. Therefore, it is little bit confusing, but you just need to remember at which stage what name is given to that structure. So again, epidermis, this we have just discussed. Epidermis, endothecium and middle layer, these are meant for protection. The tapetum, you must remember this tapetum. Tapetum is always asked in the exam. Tapetum is meant for nourishment, that is food for the pollen grain, and the sporogenous tissue is the future pollen grain. This was structure of microsporangia. Microsporangia is the whole round structure, that is microsporangia. Now pollen grain, you must have seen this in spring situation, in spring climate or summer climate. So this is what we call is pollen grain, and this was present in earlier times inside the anther. So we will see how the anther is formed, how the pollen grain is formed inside the anther, isn't it? So the anther was known as microspore inside anther, and the formation of microspore is known as microsporogenesis. This is just a fancy term in to say how the pollen grains are formed from the anther or inside the anther. See, whenever you will read the plant section of the biology, you know that biology consists of botany and zoology. Whenever you will learn the plant section, there will be more terms, and comparative to zoology, biology on the foundational stage, as botany on the foundational stage is easier than zoology, isn't it? But botany has so many terms. Let's go forward. Okay, in microsporogenesis, inside the we just discussed that the inside structure is known as sporogenous tissue. Inside the microsporangia, you see it, microsporangia or sporogenous tissue. The same thing applies. Earlier it was called at anther, and in future it will be called as pollen grains. So name is the same, but at different times the names also changes. Okay, when microsporogenesis starts, there is one pollen mother cell. Pollen mother cell, where does it come from? It comes from sporogenous tissue. You have learnt about tissues. Group of cell is known as tissue. So from sporogenous tissue, the pollen mother cells comes, and pollen mother cells undergoes meiosis one to form two cells. The two cell pollen mother cell undergoes meiosis 2 to form a 4 haploid cells. Okay, and from this microspore tetrad, many such events occur to form a group of microspores that will be in future formed pollen grain. Okay, so when a pollen formation have taken place, so that each dot what from pollen mother cell was a pollen spore, isn't it? So inside that pollen spore, it was not mature enough, so it will mature eventually after from time to time. So inside the pollen formation, there will be two vacuole will be formed. After that, one nucleus will undergo first mitosis, and then it will differentiate into two cell inside the pollen grain, isn't it, for maturation of the pollen formation. Inside pollen grain, there will be two types of cell, one vegetative cell and one generative cell, isn't it? The vegetative cell is the food for the generative cell. Okay, you will see later. Again, vegetative cell has large size. It is meant for food for the generative cell, isn't it? Now, sometimes in some plants, the pollen grain matures at two cell stage, the vegetative cell and generative cell. But sometimes the pollen grain matures into three cell stage, that is, two male gametes are formed and one vegetative cell is formed. Okay. Now, the structure of pollen, just we have seen, and we will just revise. The vegetative nucleus present is larger than the generative uh, cell, isn't it? And the generative nuclei, this is a three cell stage, has two male gametes in this case, and the vegetative has one nucleus, and this is 25 to 50 micrometers. You see micrometers, we have see 10 to the power minus 3 millimeter, isn't it? In 1 meter there is 100 centimeter. In 1 centimeter there is 10 millimeter, and in 1 millimeter, when we divide the capacity of the length of 1 millimeter into 1000 times. Are you understanding? 1 millimeter divided by 1000 times, that is 1 micrometer. See, it is very small, but when you will see in Google, and when you will eventually follow through what I am saying, you will understand. One, yeah, see, 1 kilometer is 1000 meter, isn't it? And 1 meter is 100 centimeter, and 1 centimeter is 10 millimeter, and 1 millimeter when divided in 1000 parts is 1 micrometer. So the pollen grain is 25 to 50 micrometers. Pollen grain outer structure is very hard. It is known as exine, and inner, just below exine, is present intine, made of cellulose and pectin, and it has a small pore for breathing and water absorption known as germ pore. So you just have to remember this, and you can get the details in your biology book. The Parthenium carrot grass, this is a weed. What is called a weed or contaminant is when you grow wheat on not or any other plant, you see that beside that plant many unwanted plants also grow. So any unwanted plant that grows beside your original plant is known as weed, which causes a hindrance for its growth. So that is known as weed or as contaminant. So when wheat was imported from outside, a contaminant has also come with the wheat. That was known as Parthenium. You can see carrot grass in wheat fields and in neighboring neighbors also. It is largely present, and so this from this weed or this contaminant, the pollens of this weed, that is Parthenium or carrot grass, causes pollen allergy and causes respiratory disorders. Respiratory disorders is nothing but functioning ill functioning of the trachea or the pipes present in your respiratory system. Say for asthma or bronchitis, it is inflammation or irritation of the pipes, isn't it? So the ovary, we have just discussed the male reproductive part. We will now discuss the female reproductive part. The female reproductive part, also known as ovary. See, when we were seeing the gray in the part of the flower, we were seeing the gray stick present in between, in around the green stick, isn't it? See, so the thicker stick was known as what we call, this is known as female structure or female yes, female reproductive part of a flower, and it consists of three parts: stigma, which receives the pollen grain, the style is the tube, and the ovary is the part which becomes the fruit. And inside ovary there are many smaller divisions, smaller parts, and that is known as ovules. So if you have ate pea, isn't it? Matar is known as in Hindi. So when you buy matar or when you buy pea, you see many different, you see one packet like structure and many peas, many small circle sphere marbles like sphere are present. So each marbles or each sphere is an example of ovule, isn't it? Are you getting my point? So each sphere present in the pea or each pea grain is an example of one ovule, isn't it? And the whole the packet is like the ovary, you can say, isn't it? It was we have seen this in class 11 biology: monolocular, bilocular, and trilocular, and so on. Okay, so this is the structure of female reproductive part of the flower. So we will see the structure of ovule. See, inside the ovary, the many small locules are present. Many little structure are present. Say we have just discussed that the pea is present. When you see pea, there are number of many number of sphere like structure which can be eaten, which is known as pea. So the packet is known as ovary, and each pea structure can be called as ovule. Isn't it? So now we see one structure of the ovule, which is also known as microsporangium. So where the ovule is attached to the placenta, where the ovule is attached to the ovary, is known as funicle. Above the funicle, it is known as hilum, and it has two ends: the chalazal end and the micropyle end. And inside there is embryo sac, and it has outer structures which protect it, is known as integument. And there is outer integument, there is inner integument, and nucellus is the food present inside the ovule. Again, this is just the revision. It has two ends: one is micropyle end micropylar end and the other is chalazial end. From the micropyle, from this opening, the pollen tube goes into the embryo sac for the fertilization to form the fruit. The tegument we have just discussed. It is the outer protective structure. Micropylar end is the region where micropyle is present. Micropyle is the opening from which the pollen grain enters the embryo sac, and the chalazial end, the region opposite to the micropylar end, is chalazial end, and embryo sac is located inside the nucleus. The embryo is the future fruit of the flower or the plant, which is also known as embryo sac is also known as female gametophyte. Now, female gametophyte we will see inside the ovule. When megaspore mother cell is present, okay, the megaspore mother cell you call it, or you call it embryo. It is same one word for different conditions or different situations. Okay, when megaspore mother cell undergoes megasporogenesis, it forms female gametophyte. So you remember this megaspore mother cell, and then megaspore mother cell undergoes meiosis 1 to form 2 cell, and meiosis 2 to form 4 cells, isn't it? We have learned about this in cell cycle. When a cell undergoes meiosis, it will form 4 cells, and when a cell will undergo mitosis, it will form 2 cells. You just remember about this. For cell division, we will discuss this in cell topic later on. You just remember, when a cell divides, it can divide from two process, one from mitosis and one from meiosis. When it undergoes meiosis, it forms, at the end it forms 4 cells, and when a cell undergoes mitosis, it forms 2 cells. Okay, so the megaspore mother cell, which is 2n, you just remember, when this 2n, when there is present, 2n means number of chromosomes is diploid, and when it is n, it means that the number of chromosome is half the total number of chromosome. Say for example, we in humans, there is total number of chromosomes are 46, isn't it? So the 46 is, each cell has 46 number of chromosomes, so the 46 is this 2n, isn't it? And when we talk about sperm or ova, in case of humans, the sperm and ova have chromosome number n, that is 23. Okay, so here megaspore mother cell has 2n number of chromosome, and after undergoing meiosis, it has n number of chromosome. So from these 4 structures, only one of the megaspore remains functional and other degenerate. Okay. Again, megagametogenesis. This functional megaspore, which one has survived, it undergoes mitosis and divides into 2. It again undergoes mitosis and divides into 4, and it again undergoes mitosis to form 8 nucleate stage. So this 8 nucleate stage, after the cell is 1, but number of nucleus increases, and the number of the nucleus arrange themselves in different positions. So at the chalazial end, 3 nucleus arrange themselves. At the micropylar end, 3 more nucleus forms into cell, and at the center there is 2 nuclei present. So at the end there are 3 3 6 and 1 7 cells are present. The central cell is present, having 2 nucleus, and at micropylar end there are 3 cells present, and at the chalazial end there are 3 cells present. So 3, 3, 6 and 1 in the center which has 2 nuclei is known as central cell. So 7 cell is present, but the number of nucleus is 8. We have just we will just revise that this. At the chalazal end there is 3 cell which are also known as antipodal cell, and at the micropylar end there are 3 cells, one of which is egg cells and the other are food for the egg cell that are also known as synergids. And in the central cell there is there is 2 polar nuclei, but the central cell is 1. So it is 7 cell, 3 at the micropylar end, 3 at the chalazal end, and 1 in the center is known as central cell, which has 2 nuclei. So this is also known as 7 cells but with 8 nuclei. This will be asked in the question, so pay attention and remember this diagram. So what we have learned? We have learned parts of a typical flower. Stamen is the male reproductive part which forms pollen grain, and pollen formation is from known as microsporogenesis, and what we get is pollen grain. And we have seen the female reproductive structure which is known as pistil, which contains stigma, style and ovary. Inside ovary there is ovule. We have seen the structure, and we have seen the megasporogenesis and the formation of 7-celled 8 nuclei female gametophyte, isn't it? Okay, we have seen male reproductive structure and we have seen female reproductive structure. We will now see what pollination is. Pollination is nothing but pollen grain going into stigma, and from stigma forming pollen tube, and from forming pollen tube going to the female egg or ova and forming a zygote is known as pollination. So pollination will only be said when the anther lands on the stigma. This is known as pollination only. Transfer of pollen grain from anther to the stigma or pistil, this much is only known as pollination. So pollination can be self-pollination or cross-pollination. Okay, self-pollination, what it is says that when one flower, we have seen that in a flower there is male reproductive structure and female reproductive structure. So when from male reproductive structure the pollen grain comes into the stigma and pollinate itself of the same flower, it is known as self-pollination. And the cross-pollination, pollen of different flower goes to stigma of different flower, isn't it? So cross-pollination can be of 2 types, from taking place in single plant and taking place in 2 different plants. That is, when pollen grain of one flower under what we say pollinate the stigma of another flower, but in the same plant, it is known as cross pollination. But cross pollination is of 2 types, and when the pollen grain from one flower pollinates the stigma on another flower present on the different plant, isn't it? So this is second type of cross pollination. So we have seen the self pollination of same flower, and 2 types of cross pollination present between in the same taking place in the same flower and taking place in between 2 plants. Now what we have just termed and seen the picture previously, just in previous slides, it just have given their name. When the transfer of pollen anther to stigma of the same flower takes place, it is known as autogamy. When the transfer of pollen anther to stigma of genetically similar flower from the same plant, only one plant, when cross pollination takes place, in only one plant between 2 flowers of the same plant, it is known as geitonogamy. and transfer of pollen from anther of one flower to stigma of genetically different flower in different plants, in two different plants. Okay, so the pollen is from one plant and the stigma at which the pollen lands is from second plant. Okay, so the xenogamy is taking place in two plants. Okay, and geitonogamy is taking place in the same plant but two different flowers. Okay, and autogamy is same plant and same flower. So this term will be asked in exams, so pay attention. Autogamy we have discussed. Autogamy takes place in the same flower from the same anther of the flower and from on the same stigma of the same flower. Okay, so only one flower is involved. Only one flower is involved, that is known as autogamy. Okay, okay. Now, autogamy has two situations. If now the situation, the environment is changed. Now, you autogamy can take place in the same flower. For autogamy to take in the same flower, from what the anther travels to stigma, how the anther travels, this, on the basis of this, the autogamy is of two types, chasmogamous and clastogamous. So we are talking about, whenever we are talking about chasmogamous or clastogamous, so it is involved in only one plant is involved and only one flower is involved. Okay, because this is a part of autogamy. So in chasmogamy, the anther travels to stigma by the means of any insect or bird, or just by wind it gets pollinated, isn't it? So this is known as chasmogamous. Or in chasmogamous the flower is open, and in clastogamous the flower is closed. The flower is closed, that is, the anther is very close to stigma, and when the anther becomes pollen grain, the pollen grain just fall into the stigma. Okay, you are understanding what I am saying? Anther and stigma are brought together by various mechanisms. It can be by the wind, it can be by bees, or it can be any other insect or spiders, anything. So when the anther and stigma are brought together by various mechanisms such as wind or bees or insect or spider or bird, anything, then it is known as chasmogamous flower. And clastogamous is known as when the flower is closed and anther, and when the flower is closed, the anther and stigma are very close to each other, and the anther just falls into stigma, isn't it? Okay. Geitonogamy we have discussed. The pollination takes place from anther to stigma, from one flower to another flower of the same plant. Okay, geitonogamy, you just remember, from pollen grains of anther of same flower, anther of one flower to stigma of another flower of the same plant. The plant is same, but it is two flowers are involved. Xenogamy is, in xenogamy you just remember, there is only in xenogamy there are two plants involved. The anther from one flower pollinates stigma of another plant present in another flower present in another plant, different plant. Now agents of pollination, this is easy one. You may have seen there are various types of the travels mechanism for pollen grain. It can be carried away by wind, which is known as anemophily. It can be carried away by insects, known as entomophily. It can be carried away by water. It is known as hydrophily. When it is carried by birds, it is known as ornithophily, and when it is carried by bats, it is known as chiropterophily. Biotic and abiotic agents, you understand. Biotic agents means living agents, that is bats, birds or insects, and abiotic agents is wind and water. This is same structure. Anemophily means transfer by wind. Hydrophily means pollen transfer with the help of water. Entomophily nothing but pollen transfer from anther to stigma with the help of any insect. Entomophily is entomophily is again just we have discussed in the previous slide, uh, transfer of pollen grain from anther to stigma with the help of insect. This is amorphophallus. This is 6 feet long flower. You must know and visit structures. I forgot to write down which is the location, but you just Google it. Amorphophallus. Amorphophallus is found in various places in India, and you can see go and see this flower, almost 6 feet long. 6 feet means very tall. Average human height is 5 feet 5 inch, and this flower is almost 6 feet. Ornithophily, pollen grains travel with the help of birds. Chiroptily, pollen grains travel by bats. So, okay, outbreeding devising, outbreeding devices. You just see what this term is. Outbreeding. Breeding is also known as pollination, and out means breeding is out. Okay, or in another word, stopping pollination, or stopping breeding. So you are getting my terms. So you are getting clear that what outbreeding devices is. Whatever situation, whatever methods are used for stopping pollination or stopping breeding is known as outbreeding devices. I have left one C after DEVICES. It will be there, outbreeding devices. Okay. So outbreeding devices has four methods. It is unisexuality, it is dichogamy, it is self-incompatibility, it is heterostyle. Okay, let's see. First is unisexuality. Unisexuality means flower are unisexual, and this, when flower are unisexual, means the male plant and female plant are present on different flower. Say for example, papaya plant. Whenever you grow, you must have seen papaya, papita, what we call it in Hindi, papita. You have eaten, you must have eaten. There are black colored seeds present inside the papaya fruit, isn't it? So if you want to grow papaya fruit in your vegetable garden, so you have to grow 2 or 3 or 4 5 papaya. You have to grow 2 or 3 4 5 2 3 4 5, that is number of papaya tree, isn't it? So if you grow only one papaya seed, if you take only one seed and grow papaya from only one seed, so it may be male or it may be female, isn't it? What I am saying is, if you grow only one papaya plant, it may be flower, may be female plant, or it may be male plant, isn't it? So how if you grow female plant by chance, how you should know that which seed will form a female plant and which seed will form male plant, isn't it? Say by luck you got what which by luck you sow the one seed and it turns out to be female papaya plant. So if there is no other male papaya plant nearby it, then your papaya plant will not give fruit, because the flower will not get pollinated. Why it will not get pollinated? Because only female plant is present, and no male plant is present nearby, near near it. So you have to grow 3 or 4 papaya seeds, papaya plant, that means, so that you get one male plant and one female plant. It is just a chance, it is a chance matters, isn't it? It is just a probability. You may get and may not get, but at least you have to get two plants which are of different structures, one containing male papaya plant and one containing female papaya plant, so that the male flower and female flower are present. And only in the female papaya plants, when the pollination occurs, when the pollen grain from the male papaya plant pollinates the female papaya plants, then only you will get the papaya fruit, isn't it? You are getting my thoughts. So unisexuality is a method of outbreeding, isn't it? So to stop breeding, unisexuality plays a important role. So when the male and female flowers are present on different plant, it is known as dioecious. And when the male and female flowers are present on the same plant, say for example castor or maize, you have seen that flowers of the structure and the hair present on the maize is nothing but the stigma and style. Okay, so unisexuality, how it controls the breeding by dioecious method or monoecious method. In monoecious, the male and female flower are present on the same plant, so the outbreeding is not so effective. But in dioecious plant, outbreeding is very much effective, because male and female flowers are present on different plants. Example of dioecious is papaya plant. Watermelon is also dioecious, if I am not wrong. Now dichogamy is prevent autogamy. Say for example the anther matures before the stigma of the same flower. Even if both the male reproductive structure and female reproductive structure are present on the same flower, chances are there the anther matures before the stigma of the same flower. It is known as protoandry. And when the stigma matures before the anther of the same flower, it is known as protogamy. But the space between the time period between the maturation of stigma and anther leads to outbreeding and prevents pollination, isn't it? So this is an example of outbreeding devices or stopping pollination, as you can say. Again, self-incompatibility. Sometimes the pollen grains of the flower do not germinate on the stigma of the same flower or flower of the same plant. That is, this is incompatibility, means that is not eligible. The pollen grains are not eligible for the pollination, isn't it? The pollen grains, when pollen grains are not eligible or not compatible for for pollinating the same flower or the same flower on the same plant, different flower on the same plant, it is known as self incompatibility. This prevents both autogamy and geitonogamy. This is often observed in plants of families Solanaceae and Rosaceae. Okay. Heterostyly. Heterostyly is when anther and stigma are placed at different locations. That is, the male flower in one flower only, when the stigma and anther are placed at different location. This prevents autogamy. So say if one insect comes and sits on the anther, sits on the pollen grain and does not goes to stigma, so the flower will not be pollinated. So this is known as heterostyly. artificial hybridization or pollination by humans. Artificial, the term itself means man-made, isn't it? And hybridization is pollination. So you just have to remember artificial hybridization. Artificial means by humans and hybridization by pollination. Now, artificial hybridization is crossing between two different species. Now, when sometimes when pollination takes place between two flowers of different species, say for example rose and sunflower, some this does not happen in nature, but sometimes out of luck or out of chance, the flower like rose and anther from rose, what do you say, pollinates a flower other than rose, isn't it? And some new flower is created. This is known as hybridization, that is cross between two different species. Species, what we say, species means two different genera, two different types of organism. That is, say for example, lion, tiger, and cheetah are three different species, isn't it? And rose is one species, marigold is another species, and sunflower is another species. So crossing between two different species, that is pollen from another flower and uh, stigma and ovary from another flower. So when pollination takes place between two different species, it is known as artificial hybridization. It has benefits because the benefits will be, the characters will be present of both the parents. So when characters of rose, say for rose, and say for sunflower, so the, for example, this does not happen in nature, but when this type of situation, this type of hybridization takes place, both the parental characters are shown. Okay, and this, the both the characters are shown, and sometimes development of disease resistance plants is also uh, obtained from this artificial hybridization. And when artificial hybridization is done between crops, the crops can sustain extreme temperature. Okay, you will learn this about in higher classes. And the formed what which and the new plant form is known as hybrid. Steps of hybridization, artificial hybridization. This simple step is given. So you take two flower, you can see one flower is pink in color, one flower is yellow in color. So from one flower the anther is taken and it is put on the stigma of the another flower. Okay, so the steps is emasculation. Emasculation means you take out the anther. See, the flowers of the red plant, red flower, is the anther of the red flower is plucked and making it antherless, isn't it? So that only the female flower is present in that flower. This is known as emasculation or removal of anther before dehiscence. Dehiscence is what is when anther opens up, bursts open to expose the pollen grains. It is known as dehiscence, isn't it? So emasculation is removing anther before dehiscence, so that only the female flower remains, isn't it? And bagging means bagging will be of the female reproductive structure of the flower, so that no anther gets near it and pollinates it. Again, rebagging is after after removal of pollen grain, when the time favorable conditions occur, the dust it get we make it pollinate from the we make it pollinate from anther of different flower to the stigma of different flower, and then we cover cover it. So steps of artificial hybridization can be taken in two ways: in bisexual flower and in unisexual flower. In bisexual flower, how it is done? Bisexual means when the flower contains two, what do you say, two structures, both male and female are present. Then emasculation is done. Emasculation means and removal of anther, and then bagging is done so that no pollen grain comes near the stigma, and then again pollen grain of some different flower is done, and then rebagging is done so that some other pollen grain also does not also pollinate it. And in unisexual flower, how the steps of artificial hybridization takes place is: emasculation is done, anther is removed before dehiscence, and then bagging is done. Bagging, again after bagging, the when the climatic situations are favorable, then dusting of the bag is opened and the dusting of pollen grain is done, and again it is rebagged so that it does not get pollinated from other plant. Now pollen pistil interaction: when the pollen sits on stigma, after that what happens we will see. Okay, now that the pollen grain has come sit on the stigma, if they are compatible, that is they are of same species means, say for example, if pollen grain from rose, uh, anther of the rose flower falls into the stigma of the rose flower, it will be compatible or eligible. And when the, say for example, pollen grain of rose flower falls into stigma of sunflower plant, sunflower flower, the pollen tube will not form because it is incompatible, or it is of different species, or in different species it is ineligible, isn't it? So what happens when pollen grain falls on the stigma? It forms a pollen tube, isn't it? And two cell, the vegetative cell and the male gamete transfers, makes vegetative cells makes way for the pollen tube and the male gametes transfer inside it. We just have seen that there are two male nuclei, two two generative cell. These two generative cell goes inside the embryo sac. This is known as fertilization or male and gamete fusion. So when we see fertilization, when the we have seen the structure of pollen grain, there is for in the three cell stage there is one vegetative cell and two generative cell, isn't it? So here you have seen, here you can see the generative cell, that two generative cell, the nucleus of the two generative cell comes inside the embryo sac. Okay, so when the navigation of pollen tube, the two male nuclei founds the pollen tube and the embryo sac, it discharges male gametes inside it. Okay, and one of the two generative nuclei goes to polar nuclei, and one of the generative nuclei goes to the egg cell, isn't it? So there is two fertilization, therefore it is known as double fertilization. Okay, I will revise this once again. So you just remember the structure of pollen grain. Here in this figure it has shown of the pollen grain which has three types of cell, isn't it? One vegetative cell and two generative cell. So if there is two generative cell, there will be two nucleus. So the vegetative cell forms the pollen tube, and with the help of pollen tube, see in the first figure, the navigation or transfer of pollen tube takes place, and through the pollen tube two male gametes of male gamete, that is two nucleus of the vegetative cell of the generative cell, travels inside the embryo sac. One of the generative nucleus enters and meets the polar nuclei, and the other male nucleus enters the egg cell. So there is two fusions, one with polar nuclei and generative nuclei, and another generative nuclei with egg cell. So there is, due to two type of fusion, it is known as double fertilization. Okay, that is what we have just discussed, double fertilization. The two polar nuclei fuses with one male gamete fuses with egg nucleus to form zygote, okay, at the micropylar end, and the other male gamete fuses with two polar nuclei with the primary for forming primary endosperm nucleus. This is nothing but the food for the main zygote. Okay, this endosperm is food of the zygote, food of the embryo. Okay, again, so we have seen that 7 cells, 8 nuclei stage is there, 3 at the micropylar end, 3 at the chalazal end, 3 antipodal cells are present, which are present there. And when two generative cell nuclei from generative cells come inside, the egg is one of it fertilizes the egg, this is known as syngamy, and the other goes to fuse with polar nuclei to form primary endosperm nucleus, okay, which is food for the embryo. And filiform apparatus guides the pollen tube inside the embryo sac. Types of fertilization. Okay, this is nothing but the types of formation of pollen tube. When the pollen tube enters from micropylar end, it is known as porogamy. When the pollen tube enters from chalazal end, it is known as chalazogamy. And when the pollen tube enters via the integuments or other than micropylar end or chalazal end, it is known as mesogamy. That was all for today. Let's have a quick recap on what we just learned. Okay, we will just have a quick revision on what we have seen. We have seen male reproductive structure, we have seen female reproductive structure, and we have seen what pollination is, and we have seen we will just go through these once again, a quick revision what as called. We have seen the transfer of pollen glands from anther to stigma, and this is known as pollination. And we have seen various ways to stop pollination or outbreeding devices. So this is a fairly easy chapter. You just have to pay attention. So this is sexual reproduction in flowering plants. Okay, so sexual reproduction in flowering plants, we will learn about angiosperm. Angiosperm is also known as flowering plants, is also known as angiosperm. So we have seen flowers, we have seen a typical flower. Now you can recognize what a male reproductive part is. The anther or stamen, you can say is the male reproductive part. The gynoecium is known as the female reproductive part. The thick structure in between the flower and thicker than other sticks is the stigma, or you can see gynoecium, and the thinner streaks present on a flower which are comparatively thinner than the stigma or the female reproductive structure as known as stamen. The stamen consists of two structures, the anther and the filament. Okay, this is known as male reproductive structure. When the anther is cut open,